Today we're going to see CADD and molecular modeling. A very brief but very simple and understandable introduction to these tools of rational drug design. Basically what happens is new drug discovery is a very costly, complex, competitive, intellectual, time-consuming, yet very risky process because of high failure rate and very challenging to develop new drugs. As the new drug discovery demand approaches, the aim to increase the probability of robust target identification with quick validation such that the subsequent failure rate can be reduced to manageable levels. Out of every 15,000 to 20,000 molecules synthesized and screened, only one actually reaches the clinic. And around 15,000 US dollars are required for new molecules synthesized and screened thoroughly. And therefore, the cost of developing a new drug is more than 750 million US dollars and still more increase nowadays. You can still add up another 250 million dollars. It's also challenging because approximately 15 years for a molecule to reach the actual clinical use. And in spite of this, due to heavy competition, the useful product life may not be much. It can be 7 to 10 years. Like example, we have the example of cimetidine and quinolone antibacterials, omeprazole, etc. Therefore, efforts are concentrated on cutting costs by reducing the actual number of compounds to be synthesized and screened and still achieving a better success rate. In other words, we need smarter drug design, and mechanism-based drug design is more rational and smart drug design. While discovery may be costing into millions of dollars, if it clicks, the revenues are in billions, sometimes in trillions. Let us just see the overview of the discovery process, and you can actually scale it up to 15 years like this, out of which 5 years can be given for discovery, which will involve target identification, validation, assay, screens, heats, and etc. Up to preclinical, that is animal studies and even tissue studies and all. And once you find something very interesting, investigational new drug application INDA is filed. And then the human trials begin. The phase 1, phase 2 starts with volunteers, then starts with actual patients, and the number goes on increasing, the locations go on increasing. And once you succeed in all this phase 3, you find very satisfactory, then the next process is the new drug application filing NDA, where phase 3 is really bigger one, and all side effects and all are judged. The FDA will really scrutinize it and give approval, and then after the drug is launched, and even after it is launched, phase 4 trials, post-market surveys done. So the success rates you can see, out of 5,000 to 10,000 compounds synthesized and screened, only 200 or even less can enter preclinical testing here. Others will be all rejected, out of which only 5 or even less can enter these phases, phase 1, phase 2, phase 3. And then if you're really lucky, the drug may get approved. Let us see the discovery and development. You first identify the disease, its real structure which is involved in the disease. You can call it as a receptor or whatever it is, biological structure. Then you try to find out the effective drug which can actually really modulate the disease process, and for that first you do for 1 to 3 years or up to 5 years you can find out through preclinical trials. Still don't go to the human beings. And once you find it very successful, and once you decide, you start the formulation and scale up side by side, you file an INTA. Then after human trials continue, phase 1, phase 2, phase 3, that may take up to 10 years. And if you find it very good, you file it NDA. And then if FDA approval process may take a couple of years. So in short, this is very nicely summed up new drug discovery and development process. Let us see what technology, how it impacts this process. Identify the disease, and then isolate the protein, where genomics, proteomics, and biopharm can be very useful to identify targets which are more accurate. And then through high throughput screening, you end up with large number of compounds which can be tested on these targets. Also computer-aided virtual screenings to predict the activity by doing various studies on these targets, in silico studies. Combinatorial chemistry, rapidly producing large number of compounds, okay, for lead optimization. Means from the high throughput you may find some hits, and then this can be converted to leads. And then still to refine the leads, you go for molecular modeling using computer-aided drug design, and then you do its preclinical, in vitro, in silico, also do the ADME, and then you. So here all technology comes. Let us understand what is the rational drug design is. So basically, the enzyme substrates, transmitters, enzymes, receptors, hormones are really critical links in all biochemical processes. And if one of these links is actually malfunctioning, disease or disorder occur. And modulating this malfunctioning biochemical process may alleviate or cure the symptoms of the disease. So drug targets are more often these proteins, sometimes they can be nucleic acids, enzymes, receptors, ion channels, ion transmitters, and many of them are present on cell membranes. So the mechanism-based drug design or rational drug design approach has all this taken into consideration, and efforts are to design such drugs which will target these important links and modulate them. And so it is more target based, and it helps more focused approach and helps cutting the cost, time and efforts. This is because one studies the underlying mechanism of the disease at a molecular level and in three-dimensional perspective, and all these targets, as we had discussed, are attacked by complementary fitting molecules or ligands in a very systematic way, in silico, through bioassays and other modes, and you end up with good results. You got a good guarantee of results. So this is mechanism-based drug design, most rational approach. Disease process understood. Drugs are then designed specifically to bind these disease target molecules and disrupt the disease process. It's a very complex intellectual approach and therefore requires detailed knowledge of information retrieval. And CAD becomes very handy and holds great future. The integral steps is defining the disease state or mechanism, biochemistry underlying that, defining the targets, that is receptors, enzymes, nucleic acid, and defining the effectors or ligands, that is small molecules that can fit into the active sites of these macromolecules. Let us understand that drug receptor interaction is not merely a lock and key interaction, but a dynamic and energetically favorable one. It's something like this. Though you may drug and receptor, you can equate with lock and key, but this is static and this is very dynamic. Okay. So, and in real fact, many locks are known, but not all locks are known. So you need to design properly good keys, and therefore protein crystallographic data banks give you the details of this lock. And once you understand this through various advanced techniques like X-ray crystallography, NMR, and electron microscopy, and various advanced techniques, you can actually solve these active sites and then design the ligands. So you can actually search this, and through computer algorithms you can find out the key to fitting this, which will actually fit into this properly. Okay, this is called as structure based design, or in short SBDD. So what is modeling? Many a times a word called as molecular modeling is used. And what is modeling then? Why do you need to model a molecule? In real walk of life, we hear modeling on our television also. We see a lot of models advertising things. And basically, modeling is a science that elucidates and validates experimental evidence for imagination, visualization, and rationalization. It's more effective way to express or promote something. And it's equally applied to many areas of research, particularly academic and industrial. So see, a model will be more convincing to you to understand the use or the usefulness of a particular product. Models can be human beings, models can be like coming directly to the science. DNA model will tell you more effectively than seeing it in the book, or complex receptors or proteins also can be expressed through models. Simple molecules, when we teach stereochemistry, ballistic models are more effective to explain things. And what is your periodic table chart? The properties of various atoms is nicely explained. So, why do we use models? Traditionally, physical mechanical models are used to represent the 3D molecular structure. They have a lot of disadvantages. They are heavy and must be supported by solid scaffolds. Imagine a model of DNA molecule with several hundreds of atoms, so it will be really heavy and it has to be supported with scaffolds. The flexibility and manipulation is less. Reproduction of conformation is difficult, virtually impossible to measure their atomic coordinates. Preservation problem is there. And here you understand, and here comes a terminology called as CAD, computer aided drug design. Let us see what it is and what are its advantages. Let us directly go to the advantages. Means generating models through computer and using it for drug designing is, in simple words, computer aided drug design. Let us understand its. I am going to explain you almost 10 of its advantages uh, in the next part. So definition is use of computer generated models for molecular systems under investigation to predict and explain the physicochemical properties and interaction of these systems, that is the ligand receptor. This has become a very critical component of drug design these days. It consists of several interlinked activities, namely molecular graphics, computational chemistry, statistical modeling, and molecular database management. Other words, we synonymous, or we just simply equate CAD with molecular modeling. So, as discovery is facing serious challenges because of enormous cost and time taken to discover a new drug, and also becomes because of the rigorous competition. You may work hard and you suddenly before you launch your drug, so your competitor may come out with something, so everything goes in drain. And therefore you need to be very smart and you need to smartly design. So rational drug design is a smart design which will be very effective, very focused. It will cut on your cost, time and efforts. What are the capabilities of CAD? I told you I will come to this. One can very efficiently create 3D molecular structures of both drugs as well as receptor. You can manipulate their translation, rotation, interaction, and render them in users-friendly fashion. Study their effective and favorable interactions and stability. All this can be done in real time. Moreover, one can design and predict an effective drug structure with high precision without actually synthesizing and screening it. This definitely saves a lot of cost, labor, and time. So, what are the advantages of CAD or CADD? It allows the medicinal chemist to use the computational power of computers for measurement of molecular geometry, electron densities, and electrostatic potentials, conformational analysis, and different types of energies. Direct manipulations and comparisons of key structural features of a range of biological active molecules. Generation of images through X-ray coordinates. These images can be interconverted in any forms in real time. Moreover, maps of electron population, van der Waals surfaces, volumes also can be generated. Even 3D characteristics of the receptor site can be generated, enhanced and viewed through zooming and depth viewing facilities. Different color coding of atoms, groups, molecules, and surfaces, and even chemical properties. So, more user friendly, you can really distinguish among them. Rotation and translation of molecules in real time. Docking of ligands and receptor sites, active sites, and exact measurements of geometrically and energetically favorability of such interactions. Comparison of various ligands to various parameters. So now let us get to the CAD is synonymous with molecular modeling. We saw what is modeling in general. Let us come to a more specific question: what is molecular modeling? Molecular modeling, to be very precise, comprises of four components. First is molecular graphics, that is a visual representation of molecules and the properties. Molecules can be represented by many forms as per the need, and it becomes very easy to grasp the details. Computational chemistry is actually when you are seeing all this in the heart of the CPU, lot of computations are going on using Schrodinger equations and much complicated algorithms, and to find out the exact energies and lengths, bond lengths, and so on and so forth. And so many equations are solved through the computer. So many properties are actually determined. And so this is a very complicated phenomena, and it is the heart of molecular modeling. Statistical modeling, like you do a lot of validations, QSAR, 3D QSAR, so a lot of correlations and validations, statistical tests are applied. And another very important component is information management. So much of data is generated, it is nicely sorted out. Its organization or sorting, its retrieval and search and processing of properties of thousands of compounds and comparison with potential drug candidates. So that is also very important. So in short, molecular modeling is computation, visualization, statistical modeling, and molecular data management. I think by far this is the best definition. If somebody asks you what is molecular modeling, you simply tell it's computation, visualization, statistical modeling, plus molecular data management. Not any three, all four are required. That you underline. Oh, so molecular graphics. Images are drawn by converting atomic coordinates into screen coordinates and connecting them with the lines, which are actually shown as bonds. Depth queuing of 3D for 3D effects, dimming images of atoms more distant from the viewer. Translation of images, addition, subtraction of appropriate distance along with one or more coordinate axes, and redrawing of transformed image on screen. Types of molecular images or renderings. This can be stick or wire form, most uncluttered, basically give you really which atom is connected to what. So they really give you the molecular connectivity. There is CPK, Corey, Pauling, Carlton, or spacefill models. They give you the size and shape filling for interaction for fitting, and they give all structural and steric details. Basically, are lost, but you get a rough idea of the size and actual shape. Ball and stick displays are the compromise of the above two. Mm, A dot surface displays both stereochemical details and stick figures and shape of molecular surface of interest. This can be Connelly surface, van der Waals surface, electrostatic potential surface. There are ribbon displays also, particularly useful for protein traces, and you can actually find out the amino acid backbone throughout the structure, which permits the identification of structural features, whether it is alpha helix or beta strand or connecting loops, etc. And all these which I have talked are very nicely explained uh, in the next one. So the power of molecular graphics is best shown when several of the techniques are combined to provide some real insight into the action of the drug. So you really feel, and that is in real time. So why there is a reason for explosive growth of CAD since its advent in 1980. Today it is almost 40 years old. It's really grown leaps and bounds. Development of excellent graphics display systems which can manipulate structure in real time. Tremendous increase in the speed and capacity of the hardware. Now we can do it in PCs also. Earlier we used to do with uh, workstations or super mini computers were required, but now even these ordinary computers with graphic capability can handle i5, i7 uh, chip will be sufficient. All these are coupled with substantial reduction in cost. Let us show you. This is a simple L-glutamyl residue of amino acid, and with various images how it can be. If you want to see it in stick or wire form, see, this is how it looks. If you want to see it in space fill or CPQ models, this is how it looks. If you want to see it in ball or stick diagram, this is how it looks. And as I told you, you can also see these molecules in the dotted surface diagrams. There is either electrostatic potential surface, van der Waals surfaces, or water wettable Connolly surfaces. Let us see the Connolly surfaces of this molecule, how they look. See, these are the water wettable surfaces. So you can find out the polar properties. Electrostatic potential, you can find out which is the positive electron rich and which is electron deficient part. So red are electron rich, blue are electron deficient. Van der Waals surfaces can give you a very good idea of the shape and volume of the molecule. So this is the power of molecular graphics. Now we come to the other part of the molecular modeling tool, and that is the computational tools. Computational tools are very important, like quantum mechanics and molecular mechanics. Quantum mechanics are computationally more expensive, and these are ab initio and semi-empirical methods are used. The core of this is the Schrodinger's equations, and this exclusively uh, consider the electronic effect and electron movements and electronic structure of the molecule and try to track their movements, calculate charge distribution and orbital energies, can simulate bond breaking information. However, it is so extensive that its upper atom limit is 100 atoms. Molecular mechanics are much handy, but is still little bit of accuracy is less than quantum mechanics, but it is an empirical technique and applicable to both small and molecular systems. It does not explicitly track the electron movements and calculate all these things. So some of the values of the bond energies and bond lengths and other things are directly picked up from the databases. A molecule is actually described as a series of charge points, atoms linked by springs, like a ball stick, but the sticks are like springs, flexible bonds. And the potential energy of the molecule is described by a mathematical function called as a force field. So this is how the drug works, and we have already seen last part was the statistical part, but we have already seen the uh, 3D and 2D QSAR and the statistics part that is already covered, and the data management and retrieval is really the organization. So I think I've been able to explain a little bit what is actually molecular modeling. Okay, so I hope you liked it.